Hello, everyone. I would like to thank the organizers for giving me this opportunity to present my research. Our lab primarily works on the ryanodine receptors, which are large intercellular calcium channel present on the cytoplasmic membrane. They're homotetramers, which means they're composed of 4 identical subunits. There are 3 basic isoforms of RyR 1, 2, and 3. RyR 1, the skeletal isoform, and RyR 2, the cardiac isoform, differ in their mechanism of calcium release. Membrane depolarization activates the dihydropyridine receptor, which physically couples to RyR1 and activates it, causing release of calcium from the SR into the cytoplasm. In the case of RyR2, membrane depolarization activates DHPR, causes a small amount of calcium to enter the cell. This calcium binds to and activates RyR2 and causes release of a larger pool of calcium from the SR into the cytoplasm. Most of the released calcium is recycled back into the SR via Circa pumps, while a small amount of calcium is thrown out of the cell using sodium exchangers. The vast majority of the protein makes up a large square-shaped cytoplasmic assembly with a thinner stem-like transmembrane domain. During wild-type channel opening, there is separation of subunits and significant downward movement of the cytoplasmic assembly, accompanied by a separation of the S6 helices, which opens the pore and allows the calcium to pass through it. Now, since RyR is such a large protein, it has a number of binding partners. One such binding partner is a calcium ion, which is an endogenous activator of RyR. It binds at the interface between the central domain, which is shown in green here, and the C-terminal domain, which is shown in indigo. More than 300 different types of mutations have been identified in RyR, and I have highlighted some of these in red in the Cryo-EM map in the video below. Most of these mutations are spread throughout the structure. Highlighted in yellow are the NTDA and NTDC domains of the RyR, which are going to be the primary focus of this talk. The mutations in RyR are linked to disorders such as malignant hyperthermia and central core disease, as well as CPVT and ARVD2. Some mutations are present at interdomain interactions, and so the prevalent mechanism, that is weakening of interdomain interactions, is a viable explanation. However, for other mutations which are located within a particular domain, such an obvious explanation is no longer suitable. Our aim was to determine the molecular mechanism for leakiness by single point mutants, and we used a combination of structural and functional studies to do so. We chose to study a single point mutation in NTDA, the R to C, that is arginine to cysteine, as well as a single point mutation in the NTDC, which is the Y to S or tyrosine to serine. A little bit of background on the two mutations. Both the R164C and the Y523S mutations are linked to malignant hyperthermia and central core disease, with the Y523S mutation being one of the first identified. Additionally, the Y523S mutation is also associated with heat susceptibility, which can be life-threatening. Both mutations cause a leak of calcium from the SR and an elevated calcium in the cytoplasm. The R164C mutation is less severe than the Y523S, which is linked to lethal homozygous expression. The mice carrying these mutations do not survive beyond infancy. Additionally, the Y523S mutation also shows an altered interaction with the binding partner dihydropyridine receptor, causing the activation of EC coupling to be shifted to more negative potentials. So while a lot of functional data exists explaining a lot of functional data exists characterizing these mutations, no structural explanation exists. The R164C mutation is present in a highly conserved loop, the beta-8, beta-9 loop in the N-terminal domain A. And the Y523S mutation is located in the middle of an alpha helix in the center of the N-terminal domain C. Now, in addition to the R164C, we also studied the R176Q mutation in the cardiac isoform, since it represents an equivalent loss of positive charge in a different isoform. A point to note is that neither mutation is present at interdomain interface. So an explanation such as disruption of interdomain interactions is not suitable for explaining the molecular mechanism of these mutations. Our collaborators, Dr. Murayama and Dr. Kurebayashi from Japan, generated the stably expressing cell lines for both the NTDA mutants, and they also functionally characterized them. Upon addition of an activator such as caffeine, there is a release of calcium from the SR via the RyR, by activation of the RyR, and this is seen in when the cells light up. For both mutant RyR1 and 2, the amount of calcium released was significantly lower than their wild type counterparts, and this is indicative of a depleted SR store. Ryanidine is an alkaloid that's isolated from the plant Rhynia speciosa, and it's unique because it binds to open channels. And ryanidine binding is used as an indicator of channel activity. For both mutant RyR1 and 2, the ryanidine binding was increased as compared to their wild type counterparts. Specifically for the RyR1 mutant, at very low concentrations, the binding was increased with respect to wild type. The Cryo-EM structure of the mutant channel R164C, which we determined at an overall resolution of 3.5 angstrom, indicated partial unfurling of the large cytoplasmic assembly. The three subdomains of the N-terminal domain A, B, and C move as a single entity during channel opening. Separation of the NTDA and NTDB subunit of neighboring domains was observed to be about 1 angstrom in the mutant channel under closed state condition. For context, this separation is about 6.5 angstrom when the channel opens. Along with this 1 angstrom separation, we also observed a 6-degree rocking of the NTDA domain in the mutant channel, which is shown in yellow here, versus the wild type closed channel shown in green. This is very comparable to the 8-degree rocking that is seen during wild type channel opening. So the cytoplasmic assembly of the mutant channel, which is shown in green over here, underwent a downward motion, which was very similar to the downward motion that was seen during wild type channel opening. So the cytoplasmic assembly of the mutant channel adopted a pre-activated conformation under closed state conditions. The loop containing the mutation highlighted in blue here is close to the central domain, which is a key player in channel opening. We observed a hook-like density originating from the central domain and reaching across to the NTDA in RyR1, while a similar hook-like density was observed in RyR2. It did not span across to the loop. This hook-like density was a consequence of the hydrogen bond interaction between the arginine in the central domain and a backbone glycine in the loop. And this hydrogen bond was due to the rocking and separation that I talked about earlier. Closer look at the loop itself shows that it's held together by an intricate network of hydrogen bond interactions in both mutant RyR1 as well as 2. In both structures, there is a pair of arginine residues on the opposite ends of the loop. In mutant RyR2, the arginine on the other end can reach across to the glutamine residue, whereas in the mutant RyR1, the arginine is no longer able to reach across to the smaller cysteine residue. This indicates that the shorter mutated residue has a greater effect. What's even more interesting is that the two arginine residues, 157 and 169 in RyR1 and 2, have also been linked to malignant hyperthermia and bidirectional ventricular tachycardia. In order to validate our Cryo-EM derived models, we generated reciprocal validation mutants where we changed a cysteine in RyR1 to glutamine and vice versa. The R to Q mutation actually rescued the original disease mutant and behaved similarly to wild type, whereas the R to C mutation in RyR2 was much more severe than the original R to Q mutation. A similar trend was also observed in radioligand binding assays as well as with respect to calcium sensitivity. Hence, we validated that the arginine to cysteine mutation in the beta 8 beta 9 loop of the N-terminal domain A is more detrimental than an arginine to glutamine mutation. Upon coordination of a calcium ion in the calcium binding site, which is at the interface of the central domain shown in green and the C-terminal domain shown in indigo, the C-terminal domain lifts up and comes closer to the central domain. We saw a similar lifting of the C-terminal domain in mutant RyR1 under closed state condition in the absence of any calcium. And this could be an explanation for the 15-fold increase in calcium sensitivity that was observed for the mutant channels. No such lifting or shrinking of calcium binding site was observed in mutant RyR2, and that could explain why a more modest 2-fold increase in sensitivity was observed. So the NTDA mutation R164C has a long-range domino effect which spans across several domains of RyR and is precipitated by rocking of the NTDA, formation of a molecular latch between the NTDA and the CD, as well as shrinking of the calcium binding site. Moving on to the NTDC mutant RyR1 Y523S, we observed a large downward movement of the cytoplasmic assembly shown in the video on the top. This is very comparable to the downward movement that was observed during wild type channel opening in the presence of activators. The key difference between the two was that for the mutant channel under closed state condition, the S6 helices do not pull apart and the pore remains closed, whereas for the wild type channel, the helix is separate. In order to numerically quantify the large scale movement that we observed, we generated rotation and translational values for each individual domain of the RyR and generated a heat map, which we then projected onto the Cryo-EM maps. You can see that for the mutant channel under closed state condition, vast majority of the domains of the cytoplasmic assembly have already undergone relocations that are comparable to comparable to wild type channel opening. For the mutant channel under open state conditions, some of the domains of the cytoplasmic assembly have in fact moved more than they would as compared to wild type channel opening. Highlighted in red boxes here are the N-terminal domain C, the domain which contains the mutation of interest. In order to better visualize the movement that we saw, we also calculated vector arrows which show the directionality of the movement. Comparing the mutant and the wild type channel both under closed state conditions, you can see that the cytoplasmic assembly shows a large conformational change. The transmembrane domain shows changes of a smaller magnitude. Comparing the panel on the left with that on the right, which is conformational changes that the channel undergoes upon addition of activators such as ATP and calcium, you can appreciate that the single point mutation is indeed causing a large conformational change. The only difference between the two, only major difference between the two is that the S6 helices in the mutant channel remain closed. On addition of activators, calcium and ATP, the mutant channel opens up. Since the mutant channel has already undergone such a large conformational change, very little is required to fully open the channel. Comparing the wild type and the mutant channel, both under open state condition in the presence of activators and ATP and calcium, shows that the mutant channel undergoes further movement as compared to the wild type channel, both in the cytoplasmic assembly as well as in the transmembrane. Taking a closer look at the N-terminal domain C, the domain containing the mutation of interest, it lies in the center of the NTDC and is cocooned by 4 helices of the NTDC. The NTDC itself is in proximity to the central domain, which interacts with the C-terminal domain. The interface of the CD and CTD is where the calcium coordinates. You can see on the morph on the left over here that the rocking of the NTDC is transmitted to the central domain and causes a lifting of the C-terminal domain towards the CD and a shrinking of the calcium binding site. This lifting of the CTD is comparable to the movement that it undergoes during wild type channel opening. Perhaps is better appreciated by these distances. In wild type channel, the distance is about 10.1 angstrom and it reduces to 8.1 upon coordination of a calcium ion. In the mutant channel, in the absence of any calcium ions, the distance shrinks to about 8.9 angstrom. In the mutant channel under open state condition, we see a clear density for the calcium ion and the distance decreases to 8.2 angstrom, which is comparable to wild type open. The radioligand binding curve over here in green represents that of the mutant channel. You can see that at very low calcium concentrations, the mutant is shows increased binding to ryanodine as compared to wild type, which is shown in black over here. Perhaps this is better appreciated in the bar graph here, where you can see that the Y523S mutation shows increased binding as compared to wild type. And also see that the R164C mutation shows a more modest increase in binding as compared to wild type. Just as a reminder, we determined the structure of the mutant channel under closed state condition, that is 2 millimolar EGDA, and under open state condition, that is 1.5 micromolar calcium and 5 millimolar of ATP. Already published single channel recording for this particular mutant shows that in the presence of low concentrations of calcium 6 to 8 micromolar, the channel open probability ranges anywhere between 0.2 to up to 0.95 and is significantly higher than that of wild type. Not only is the channel open probability increased, but also the channel open time is increased for the mutant as compared to wild type. A very detailed analysis of the pore profile shows that the mutation itself has a marginal effect on the pore at both the hydrophobic gate as well as the second constriction point. The S6 helices of the mutant channel remain closed. Upon addition of the activator calcium and ATP, the channels open up, and we see an additional dilation at the hydrophobic gate 4937. Shown on the right here is a pore profile for those structural biologists in the audience. So far, I have been speaking mostly about the global and the large-scale conformational changes in the large cytoplasmic assembly of the channel. I'm going to focus a little more on the molecular mechanism that underlines the mutation. The mutation is located in the core of the NTDC, and you can see that the entire of the NTDC rocks during channel opening as a single entity, and that the individual helices remain static. Whereas in the mutant channel, the helices undergo rearrangement, specifically helix H6 and H7, which forms a bent king helix, undergoes a inward movement of about 1.3 angstrom towards H8 and H5, which changes the angle of this helix. Shown in ball and stick here is the tyrosine residue and the serine residue of interest. This rearrangement, which is perhaps better appreciated when I make the other helices transparent, so that you can focus on the inward movement of the H6, H7 helix towards the H8. This rearrangement, which is seen in the mutant channel, is absent in the wild type channel during opening. So all of these changes in the core of the NTDC, the rearrangement of the alpha helices, etc., are transmitted to the central domain via the H3 helix of the NTDC, which is in direct contact with the alpha 2 helix of the central domain. This interface between the two helices from the two different domains is dominated by polar interactions, and these interactions are present in both wild type closed as well as open channel also. So about a 5-degree rocking of the N-terminal domain is conveyed to the central domain, which results in a lifting of the C-terminal domain and a shrinking of the calcium. We discovered a rigid lever about 100 angstrom in length connecting the DHPR facing surface of the protein to the interior of the protein very close to the calcium binding site. The conformation of this lever, which is composed of 2 alpha helices from NTDC and 2 alpha helices from the central domain, is altered in mutant RyR1 and might explain the change in RyR1 DHPR couple. In conclusion, we have determined cryo-EM structures of NTDA and NTDC mutants. For the NTDA mutant, arginine to cysteine mutation in the beta-8, beta-9 loop was more detrimental than an arginine to glutamine mutation. The mutation caused the channel to adopt a pre-activated conformation, and the calcium binding site of the mutant channel was altered, perhaps allowing easier coordination of the calcium ion. For the NTDC mutant, tyrosine 523 acted as a spacer residue. The extent of pre-activation observed was much larger, and this cryo-EM observation is in agreement with already published functional data. The calcium binding site of the Y523S mutation is also altered in the absence of activators, similar to R164C. So while both mutations show a common domino-like effect, the exact molecular mechanism by which they achieve this effect is distinct. And this highlights the significance of studying these single point mutations in the context of full length RyR. I'd like to thank my advisor, Dr. Samso, my colleagues, Dr. Nayak and Dr. Hugh. A special thank you to Dr. Elteet for teaching me basics of cell culture as well as live cell calcium imaging. A thank you to our collaborators, Dr. Murayama and Kurebayashi, who generated the cell line and functionally characterized the mutants. I want to thank Thomas and Valerie at Purdue and Adam and Thomas at NCI for all of their help during data collection. Also would like to acknowledge the funding sources as well as BCU, and I would be more than happy to answer any questions. Thank you.